It's also one of the leading research programs in the country. They actually, Center for Life Sciences is actually headed by Dr. Aynur Akiljanova, and she's also head of genomics and personalized medicine laboratory within CLS. So we have 8 laboratories, and they're all actually focused on high-impact research to advance science and improve healthcare and well-being of citizens of Kazakhstan. So I don't go to every detail because most of our speakers today from CLS will present their research projects. And I would like to say that our vision is to become one of the leading research organizations, and I think within and internationally recognized. That's why we're working on it. And our mission is, of course, is conducting high-impact research, and today we would like to really share all our major advances in our research programs and share our new ideas and thoughts. And we together, I think, when we hear from you within these 3 days, I think we come up with a major new ideas how to work together and maybe build strong collaboration. And that's the main idea behind this, our research conference. And technically, NLA is doing pretty good by funding, getting major funds from Ministry of Science, Higher Education, and also, of course, with support of Nazarbayev University. We're having around annually more than $10 million per year. I think that's good research funds for developing our programs further. But we're looking forward to enhance and broaden our research programs. So we are also trying to publish, improve our publications, high-impact journals, and we are targeting now, of course, Q1 publication. And one of the main things is that we are together with the university. Our main focus is to move forward with our ranking with time and higher education within 30 percentiles. That was a major achievement for Nazarbayev University. Within 3-4 years, we are targeting to get to 20 percentile. I think and research is one of the instrumental part of it, and together we will get there. I think that's our task. I think that's what the evidence of it, of course, we have to publish better papers and deliver high impact. And so thank you for your participation in our major event of the year. Thank you very much. Hoş geldiniz, welcome. And thank you for, now it's time, actually, now I would like to introduce our next speaker is actually President of Nazarbayev University, Dr. Waqar Ahmad. Thank you very much, please.
Well, welcome. 8:30 as a start for a conference. There are not many conferences which will start at 8:30. And it seems that some of the delegates are still asleep. We have quite a few empty seats. So hopefully during the day, this hall will fill up. It's an honor and a pleasure for me to welcome you to this International Scientific Conference, Life Sciences Today, Frontiers in Biomedicine, Infer, Innovate, and Impact, organized by the NLA, which is so ably headed by Dr. Dos and supported by a team of very eminent scholars and researchers in this country. This is one of about 20 conferences that we have funded this year. We funded these conferences not because we like organizing lots of events, but because we have a special role in this country. We have a role as a catalyst for reforms in higher education and for developing Kazakhstan as a knowledge economy. We're a leading university. We take that role very, very seriously, and through these conferences, we have provided a platform for the rest of the universities in the country and researchers elsewhere to come and enter into dialogue, debate, develop partnerships with fantastic researchers from across the globe. So this is one of those many events, but a very significant one, and it's a pleasure to have you here. As Dr. Dos said, this is the 15th anniversary, both of the university and of the Center for Life Sciences, and the 10th anniversary of National Laboratory Astana. Life sciences today stand at the forefront of discovery and transformation. I am not a biological scientist, I'm a social scientist, but a lot of my work has been on issues to do with health and illness, so from time to time I have worked with medics and life scientists. And the role that you play goes from deepening our understanding of the human genome and molecular mechanisms of disease to advancing translational research that bridges the laboratory and the clinical practice. This field is shaping the future of medicine and public health. And it's particularly important for us as a university. We have this fantastic center. We have great research in biology and biomedical sciences across the university. But we also have a medical school where we teach, and we have 4 teaching hospitals. So this triangle of relationships between teaching, research, and clinical practice gives us a model for integrated healthcare, which we're very keen to export to other universities across the country and across other providers across the country. Pardon me. For the past 15 years, the Center for Life Sciences has been a hub of innovation in genomics, bioinformatics, biotechnology, oncology, microbiome research, and bioengineering. Its contributions have strengthened the scientific foundation of Kazakhstan while opening new avenues for collaboration across the globe. The NLA, in turn, has become a platform where interdisciplinary ideas are born, international partnerships flourish, and innovations find their way into practice. With its advanced infrastructure, talented researchers, and strong ambition, NLA plays a vital role in building a knowledge-driven economy and positioning Kazakhstan as an active player in the global scientific community. This conference is more than a scientific forum. It is an invitation to dialogue between fundamental science and clinical application, between academia and industry, and between Kazakhstan and the international community involved in research in these areas. Such exchanges accelerate the journey from discovery to real-world solutions, from the bench to the bedside, as Dr. Dos and others would say, and bring us closer to shaping a healthier and more sustainable future. I wish to express my sincere gratitude to the organizing team. Dr. Dos, I'm not sure where he gets the energy, given how many things he's involved in. He does all of them well. My thanks also to our distinguished speakers and to all of you participants, and thank you for waking up early and getting here well before half past 8. Pardon me. Special thanks go to the dedicated colleagues of the Center for Life Sciences and the National Laboratory Astana, whose tireless efforts have made this event possible and continue to advance science in Kazakhstan. Dr. Dos mentioned our ambitions as a university, and I say to people that we are more than a university. We carry the aspirations of this fantastic, new, bold, precocious country, which wants to be counted amongst the community of nations for the contribution it's making to the development of science, the development of technology, and the betterment of society and the environment. And so our role is not simply teaching, it's not simply research, but it's also uplifting the higher education and research across the country. It's a role that we take very, very seriously. So finally, I wish you all productive discussions over the next couple of days. I may pop back in again if there's time. I think I'm slated in to join you for dinner at the end of the conference. So I have a wonderful couple of days, and I'm sure that your discussions and deliberations would be of benefit not just to the NLA, but to the community of researchers in this area more broadly. Thank you.
Very much, Dr. Ahmad. So and now it's time for really to introduce next speaker. Is actually with my all full gratitude, I would like to invite to the podium the founder of Center for Life Sciences is Dr. Zhaxybay Zhumadilov. Please, thank you. Please come.
Құрметті әріптестер, замандастар, бүгін өте бір ерекше күн, үлкен мереке, осы мереке құтты болсын. Сіздерге айтайын дегенім, ең бірінші денсаулықтарыңыз зор болсын. Шаңырақтарыңыз биік болсын, бақ береке болсын ішінде. Құтты болсын мереке. Dear colleagues, friends, it's my great pleasure and honor to make this brief speech to congratulate my colleagues with 15th anniversary of the university. I wish you all the best, and let's enjoy great research outcomes. Ну, говорят, время проходит, уходит, но получается на деле мы должны догонять это время для того, чтобы успеть выполнить свои планы, для того, чтобы внедрить их, что очень важно. Надо сказать, что сегодня Life Science Center публикуется в престижных международных журналах. Это Lancet, Nature. То есть это то, о чем раньше мы могли только мечтать. Сейчас это стало традицией публиковаться именно в престижных журналах. Поэтому я хочу пожелать вам дальнейших творческих успехов. Спасибо.
Ортаға тұрыңыз. You are not going easy, straight out, so please, we really have to come, all our colleagues. We have to show our appreciation, and we actually would like to provide a certificate of appreciation. It's presented to Zhaxybay Zhumadilov in recognition. In recognition of your outstanding vision, leadership, and dedication in establishing the Center for Life Sciences, which has become a foundation for advancing science, innovation, and education. Your invaluable contribution will have a lasting impact on the scientific community and future generations. Көп рахмет. And with that, yeah, we also have a Sharman, as the Kazakh tradition, to our founder, Zhaxybay, Zhumadilov. Thank you, I greatly appreciate it. We look forward to hear more from you. Thank you very much. Көп рахмет сізге. Thank you. So, it's a great moment. Thank you very much. Zhaxybay, that you came to really open up our conference today. And now I would like to also introduce founder of Center for Life Sciences and one of the major contributor in establishing Nazarbayev University is academic member, Professor Almaz Sharman. Please, thank you very much. Come.
Құрметті қауым, әріптестер, достар, бауырлар, бүгін маған өте маңызды күн. Мен өте қуаныштымын, бүгін енді 15 жылдықты біз бірге осылай жиналып тойлағанымызға. Әлі күнге мен есімде 15 жыл бұрын бірінші осындай жиналысқа біз бәріміз қосылып, сырттан біраз мамандар, керемет ғалымдар келіп, солардан біраз білім алып, ары қарай өздерің жалғасасыңдар. Мінекей біздің бүгінгі орталығымыз қандай керемет жағдайға биік деңгейге осылай шығып, мен өте кереметтей қуаныштымын. Сіздерге мың бір рахмет, осы бәрін қолдап осылай жүргізгендеріңе. Dear colleagues, it's my pleasure to welcome you this important event, this 15th anniversary conference on life sciences. It's a great honor, of course, to participate in this conference. As Dos mentioned, that I'm one of the founders, but I personally would like to pay tribute to my friend and colleague, Professor Zhumadilov. Thanks to his wisdom, thanks to his knowledge and experience, we managed to establish such a formidable team of very motivated, ambitious researchers who brought the Center for Life Sciences and now NLA at such a high level. And I'm very proud of these achievements and accomplishments. And thanks to 15 years ago, thanks to our collective far-sighted judgment, I would say, we selected 4 areas of research that we thought at that time would be probably the most promising, and those were Center for Personalized Medicine, Center for Regenerative Medicine, Center for Longevity Research, and other areas. And now they evolved into centers for bioengineering, bioinformatics. So this life that, things evolving, and now with the advent of artificial intelligence, I wouldn't be surprised that there will be even more important areas that may bring us better understanding of health and healthy longevity. I remember many faces, those young researchers who just came to our lab 15 years ago, and now I can see that you became really accomplished scientists with great publications at very prestigious journals. And I wish you all the best in your important, very important endeavor. And we believe that we are very fortunate living in such time when so many things happening, and I believe that we we now at the advent of many more discoveries that would help people to preserve their health, restore the health, and to live long and happy life. Сізге мың да бір рахмет. Бүгінгі жиналысқа келгендеріңізге сәттілік тілеймін, жақсылық тілеймін, зор денсаулық. Рахмет сіздерге.
We learned from you. Yeah, please. So it's also a great pleasure to provide a certificate of appreciation is presented to Professor Almaz Sharman for being the inspirational mind and initiator behind the establishment of the Center for Life Sciences. Your dedication to advancing knowledge and your unwavering belief in the importance of life sciences have provided the spark that made this achievement possible. Your vision continues to motivate and guide the scientific community toward new horizons. Thank you very much. Thank you. Thank you very much.
Можно я селфи сделаю?
So we're continuing. And now we would like to invite actually next speaker is actually to make remarks on for Center for Life Sciences. Actually, you could say heavy artillery, top scientists of Kazakhstan who are actually driving science and who is establishing major programs in genetics. It's actually my professor, academy member, and Professor Rakhmetkazhi Bersimbay. Please welcome. Yeah.
Құрметті бүгінгі конференцияға жиналған қауым, мен де қазақшадан бастайын. Мен де өте қуаныштымын. Неге десеңіздер, жаңа Зhaxybay Zhumadilov айтты ғой, біз сол кезде осы университеттің жаңағы басталып істеп жатқан кезінде мен де араластым, сол кісілер барлығы жасағанда ойлайтұғымыз, мына біздің жастар қай уақытта осындай жетістіктерге жетеді екен деп. Құдайға шүкір, бүгін міне сол жетістіктерге жетіп, біз үлкен жаңалықтарды ашуға өздерің бірге жұмыс істеп жатырсыңдар. Барлықтарыңды осы үлкен мерекемен құттықтаймын. Чтобы сократить время, я тоже написал, университеттің ректоры сөйледі ғой, сонда мен қазақша, орысша, ағылшынша жазып әкелдім. Сонымды уақытты созбай үшін соны айтайын. Dear colleagues, first of all, I would like to congratulate all members of these two centers, Life Science Center and the National Laboratory of Astana, with these two anniversaries, 10 years and 15 years of these two research laboratories. 10 and 15 years, it is not very long time, especially from the point of time in science. But during this very short time, you made a lot of brilliant researches and you published your researches in different very prestigious, very well-known journals. and I wish you all the best in future work. This conference is dedicated to the 15th anniversary of the Center of Life Science of Nazarbayev University and 10th anniversary of National Laboratory of Astana. This conference will be a major event for both basic researchers and scientists and clinicians who are involved in the development of new innovative areas of molecular medicine. Currently, the world is actively developing cell technologies and genetic engineering, new methods, gene and genome editing, research in the field of stem cells, and wide genomic and proteomic researches. These areas have become the basis for personalized medicine that in the future can be used to treat the most severe diseases. In many aspects in the near future, you will receive mainly cancer patients will receive the drug for treatment of this disease. I wish your young team new ideas, new achievements of scientific goals, new horizons in the most interesting field of knowledge. I wish all conference participants fruitful discussions and great scientific success. Thank you.
So, one more remarks I would like to hear from Professor and member of Academy of Sciences of Kazakhstan, Yerlan Turuspekov. Yeah. Welcome. Thank you very much.
Good morning, ladies and gentlemen. First of all, thank you very much for the organizers of this wonderful conference, wonderful event in our community. And I think it's very important. I look through the agenda of the conference, and I think that it's full of fantastic talks provided by outstanding scientists from many well-established organizations all over the world. Therefore, I think that it is already a big success, and I'm thankful for the organizers, and I'm confident that the conference will help us to work together and discuss many important issues. And most of all, I think that it's an exciting event for our young generation scientists that will be gaining a lot of knowledge from attending this conference. Well, the Nazarbayev University is a true leader in our research community and working in many areas in aspects of science in Kazakhstan. Just for example, this year we established Kazakh Society of Geneticists and Breeders, and the society consists from 4 sections, including human genetics section led by Professor Rakhmetkazhi, and the university has done a fantastic job for the initiation of these activities. And as a chairman of this society, I hope that we work together hard to develop genetics in our country. Well, I'm just sincerely congratulate the entire community of Nazarbayev University with these anniversaries, and I think that the work done here in the Center for Life Sciences in the National Laboratory Astana is fantastic, tremendous work, with lots of scientific achievements and discoveries. And I hope and I wish you a bright and long-lasting future. Unfortunately, the president of our Academy of Sciences couldn't come because of his duties for other events, but he sent a greetings letter full of warm words here. And I just, to make my mission accomplished, will read a few sentences, final sentences from his letter, and it's written in Kazakh language. Қазақстан Республикасының ұлттық ғылым академиясы сіздердің ғылымды дамытуға қосқан үлестеріңіз үшін шынайы алғысын білдіреді және конференцияның нәтижелері еліміздің ғылыми әлеуетін нығайтуға, сондай-ақ биомедициналық зерттеулердің әлемдік деңгейде одан әрі дамуына ықпал ететініне сенімдіміз. Сіздерге жемісті еңбек және идеялар мен ғылым денсаулық сақтау және қоғам игілігі жолындағы маңызды жаңалықтар тілейміз. Ізгі ниетпен Академия президенті Ақылбек Күрішбаев. Көп рахмет. Спасибо. Успехов, коллеги.
I think now, thank you. We have one more event to take care of. It's actually picture time, and then we'll start science. So please, how we'll just gather, right? I think, who's our, how we'll do this, yeah? We have to stand this way?
You should stand and...
Please stand, and then you might pick up better seat. Please come here to the podium, and then we have to take picture. We'll be taking from the wall, yeah, side. We have to. Yeah, it's just one more thing. Let's take pictures before we. Because some people have to leave, we would like to really, it's a historic moment to be sure we all have. Yeah, please come, yeah. We have also. Yeah, Dave, Yasmin, please come, yeah, don't be shy.
Доктор Махачев, вы где встанете? Здесь?
Я там встану, я сейчас. Как у вас, нормально?
Не надо туда идти сюда.
Коллеги, пропускаем вперед. Секундочку. Да, быстрее, чем обычно.
Юля, это вам, да? Юля.
this is. Yeah, please take your seats. It's only now we're getting started. It was just a warm up. So, Yulia, please take your time. We have many things to take care today. And I feel like I better walk around because I really don't see the screen. Welcome. We're back to celebrating 15 years of innovation. At the Center for Life Sciences, from discovery to the bedside. And I had only 15 minutes to present all our achievement or like major research achievements or our current state of Center for Life Sciences. But then I realized because it's of time limitation, I decided just to show you one example how we can do from discovery have some impact to bedside. And I would like to present, and this is what we do at Center for Life Sciences. We have a variety of projects from genomics, bioengineering, stem cells, and microbiome, cell migration, variety of broad area of research. And of course, we look forward through advancing science, also improve healthcare and well-being, as I said. So I'd like to present one of my research topic is defining the nutrient-dependent regulation of protein synthesis mediating cell survival in cancer cells. So that's one of the areas we're pushing as a basic science, but I feel we can get to the major breakthrough. So this slide comes from David Sabatini that when I was in his lab, I really like this slide. We show fundamental research, cell growth, what is required. Nutrients are essential for regulation of driving cell growth. And through finding this compound rapamycin, major field of mTOR was discovered. And when I was at MD Anderson, one thing I turned around actually, I was looking which nutrients are essential for cancer cells. What was the finding, of course, is not a surprise that for highly malignant cancer cells, KRAS mutant cancers were most sensitive to glucose deprivation, not amino acid. And when I saw this, I realized maybe I would like to study how they die. It was at like around 10 years ago at MD Anderson. So, and what we found is that they die, mostly KRAS mutant is not a targetable oncogenic driver, major one that is really common, drives major highly malignant cancers, and it's not still targetable. Yeah, so cells during under glucose deprivation, they die because of ROS production. What cells do, they produce a lot of ROS by mitochondria. And what I realized when I saw this, came up with the idea, maybe I cannot do glucose deprivation, but what we can do directly target this by oxidants. And at that time, we started using very simple, try to see arsenic trioxide is a drug that was already a common drug used in treat leukemias. So, and what we see is on the right that ATO is actually somehow can affect this ROS production and kill cancer cells, but at high dose. But then Luke Antley at that time came up with major findings that vitamin C at high concentration, high dose can also induce oxidative stress. And when I put all these things together, I realized I can use these two drugs to target KRAS mutant cancers, and it appears to be pretty potent. So you can see on the right, you can see that a combination is highly potent to kill KRAS mutant cancers. Not alone, drug, DVC, or other. Even D-form vitamin C was highly effective, and they all die through ROS production. It was highly effective also suppressing tumor growth as well, so suppressing tumors in xenograft model. And now, when I got back to Kazakhstan, we were ready to push clinical trials because vitamin C D form is actually FDA-approved compound, and arsenic trioxide is already used as a drug. So we started first 2022. I had to really convince medical doctors that really arsenic can be used because it was not available in Kazakhstan at that time. And we started with low dose, about 0.15 gram per kg of vitamin C, D form, and the standard dose of arsenic trioxide. So in 2023, actually, we came up. The good thing, we actually realized that there was some effect in second phase. Patients at the group, so they started having stabilizing tumors, so the tumor growth was not advancing. And one patient has even suppression of tumor, about 30%. And that was only beginning. Actually, later, actually, we got more funding to follow this drug combination, and now we are pushing this 2024, 2026, and we try to increase DVC up to 0.6 gram per kg. But I think it's still not as effective because arsenic is still behind. So, and we spend a lot of time to preclinical studies to show that we can increase arsenic trioxide up to 0.5 milligram per kg. And I think now we finish our clinical studies, preclinical studies in mouse and rat model. So, and now we are actually trying to get approval and push this study further. So that's where in our clinical trials, I think we have to finish them next year. So with that, as I'm telling, we're back, but we still, of course, we don't do only clinical trials, we do some basic research. And one of the areas we still push, of course, my favorite pathway, mTOR, target of rapamycin, that control growth and accumulation cellular mass. These two complexes were identified while I was in David Sabatini's lab, and that was really advanced field of understanding how growth of cells and proliferation controlled by this essential conserved pathway. So there are, if you look at it, this is mTOR controls protein synthesis, cell growth, cell proliferation through second complex, but there are also some nuclear anabolic processes that also controlled by mTOR, but they are poorly characterized. And of course, one of them is still remained as a big puzzle is ribosomal biogenesis. So, and our interest was to define how actually this pre-ribosomes get formed, and I think this area is very exciting because it takes about 2 hours to assemble full ribosomes, and there are not only putting proteins, ribosomal proteins, about 80 proteins with ribosomal RNAs. There are also, as I would like to see on here, it's actually ribosomal RNA modifications are very critical, like pseudourydylation, acetylation, and others, so, and methylation. So they're very critical for the function of ribosomes. And when we learn how to do pre-ribosomes, we realize when we starve cells, pre-ribosomes are still made. That was something that were unexpected. They made even higher rate higher pre-ribosomes compared with starving condition. In blue is the control, red is minus amino acid, and in black is minus serum conditions. So that was surprising, but beside, what we also see, we know that cells respond very well. When you starve them, they stop proliferating. There's not a surprise. But what we know, they do not die. Cancer cells do not die if you starve them. Serum deprivation or amino acids, only glucose deprivation, KRAS mutants start dying after 48 hours, but they actually don't die for a long time. And there's definitely adaptation takes place. So polysomes, if you run a ribosomal polysomal profile, you can see that under minus serum starvation, there is definitely polysomes maintained pretty high. Even under minus amino acid, we see some decrease, but there's still pretty substantial amount of polysomes. So there is definitely major adaptation takes place. And what we study is, and we realize, can we see what the profile transcriptomics can tell us? And what we see is that under minus serum conditions, we see that fatty acid synthase pathway and also cholesterol biosynthesis are getting genes that involved in this pathway, they get upregulated. Now it's kind of reasonable because without serum, there is no lipids are not available. And that's why they have to make their own lipids. And what is good is if you block synthesis of cholesterol by simvastatin or cis-75 fatty acids, so cells start dying under minus serum conditions. That was also just beginning. We actually could predict this, but that was really showing that functional transcriptomics is working. And our main interest, of course, to understand what's happening under minus amino acid starvation, how they adapt, because polysomes are still high. And what we see is that when we did transcriptomics, total mRNA and polysomal mRNA, so we see there are compared about major upregulation takes place. 2,600 genes are going up, and they're in total RNA and also in polysome fractions. So our working model is, then when we of course did transcriptomic, we realized unexpectedly during amino acid starvation, our main pathways that get upregulated are related to ribosomal biogenesis. This was very unpredictable, and mRNA ribosomal processing, ribosomal RNA metabolic processes, maturation of ribosomes. So it means there is a major change. So it means ribosomes made actively, but they are probably different. And because they have to survive under amino acid starvation. So we got back to our model, and what is our working model is most likely there are new ribosomes are made, and not like very resilient ribosomes that work under minus amino acid conditions. They probably selectively translate a subset of mRNAs efficiently, and it happens, of course, through difference in modification ribosome can happen through different modification ribosomal RNAs, and that's what we're pushing. And besides, there is also some specific set of mRNAs can also be modified and post-transcript. So we also, by different modifications, and that's what we are right now. But the problem is there is a major gap in this field because post-translational RNA modifications are still poorly characterized because there is no good methods to study that. And of course, now actually what we realized, there is a major breakthrough in technology. It's called Oxford nanopore sequencing, when pore directly sequence DNA or RNA. And thank you for Nazarbayev University, actually, who provided our major tool and platform of ONT and Illumina also. But we realized we can push this project directly sequencing, and through direct sequencing, we also you can see when pore can sequence directly, they can also detect some modifications. And this is one of our most exciting project right now. We are pushing this, I think, with advancing ONT right now. Recent platform of Promethean is allowing to catch these modifications. And that's where we are right now. We're trying to gather as much information and come up with a major understanding how cells adapt to some starvation conditions. So that's what we're going. That's I said. So we're trying to understand through how cells adapt, through studying how ribosomes are made, how they change, which kind of modifications takes place, how mRNA can get modified, and which proteins are actively get synthesized. And this is acknowledgement slide. I really greatly appreciate Dr. team of oncologists from Kazakhstan that really pushing our clinical trials. Another lab members at C4, that's all my grad students, and Agata Bursa, who became also as a faculty at Nazarbayev School of Medicine, and NLA also great support. And major first funding comes from Ministry of Science and Higher Education, Nazarbayev University. Thank you very much, and I look forward to discuss all these ideas I was sharing with you. So do you have time for? No, I mean, there is no time for questions right now. How much? So now, Professor Almaz Sharman, please, you're also moderator today. Please join, yeah? Sure. And so because we slightly delayed our start, and I think we'll have time to discuss. I know we have many colleagues here that probably some ideas getting very crazy, but I think life is life, so cells are resilient, they adapt very well. And now I would like to actually invite our second speaker this session, Professor Almaz Sharman, President of for Academy of Preventive Medicine, Kazakhstan. Thank you.
Thank you, Dos, for introduction and for the nice presentation. So if Dos presented about how to starve cells, I'll try to present how to better nurture human in order to become healthy and live longer life. For most of modern medicine, we've been reactive, waiting for symptoms when treating disease. But the new frontier is proactive medicine, predictive risks, spotting disease before it starts and acting earlier. Today I'll walk through how that shift is happening across heart disease, cancer, brain cells, and metabolism using biomarkers, artificial intelligence, and lifestyle interventions. So think of it as moving from sick care to true health care. So here's the framework of what's called proactive medicine. I don't know. Here we go. So the framework for what they call proactive medicine, the first is forecast risk early before symptom starts. Second is detect organ damage before symptoms. Third is deliver the right action at the right time. So if we do those three things, prevention becomes precise and personal, not guesswork as we usually practice. So the engine, the technology behind all of this is primarily the data. Genetics tell us inherited risks, proteomics show us what's happening in real time, and organ clocks reveal biological wear and tear. And artificial intelligence put all of this together, building individual health timelines so we know when and at what stage of life we should act. So artificial intelligence is very fashionable term these days, but here it has very, very specific meaning. It means large language models, but it doesn't mean that we have to invent new LLMs because the LLMs that are widely used in medical field are quite available and they are very sophisticated. It's MedPalm. So we can also use open source LLMs such as Llama, Mistral, Quen, and also use RAG for medical resources. So what we really need is to use existing large language models and fine-tune them, train them using existing information that's available in the medical field. But what's important is integration of everything into a medical IT ecosystem, and that's a key. So we can combine voice, text, data analysis, report, and create health passport. That's what we currently do on routinely basis in our clinic in Almaty. So let's start with heart disease. We know that low-density lipoprotein cholesterol and ApoB are causal in the development of atherosclerosis. So in this case, the target is clear. So we reduce low-density lipoprotein cholesterol to 55 milligram per deciliter, and we can really improve health and reduce the risk of many, many cardiovascular diseases, especially if there is a plaque discovered or identified in the coronary artery using ultrasound, which is very, very common now, especially in Kazakh population after the age 55. What is important also is high sensitivity as C-reactive protein. So we routinely use CRP as a just routine blood test, but what's important is in order to determine chronic inflammation, high sensitivity C-reactive protein are very, very important, and along with ApoB and lipoprotein A, what's called small lipoprotein. So all of this help us to determine what are the risks of cardiovascular events, and this basically inform us about what the best intervention. So on the top of that is once we discover those risk factors for atherosclerosis, we can engage anti-inflammatory strategies. And of course, this is diet, exercise, everybody knows about. But what's lately known, it's a low-dose colchicine is also very important to reduce. It's a 0.1 milligram of colchicine now becoming very, very there's compelling evidence that really help to reduce inflammation. Plus, lately there are a lot of studies that demonstrated the GLP-1 agonists, such as Ozempic, but nowadays it's Wegovy and Zepbound and such drugs. They help to reduce inflammation and certainly reduce the risk of cardiovascular events. Cancer is very important. I think at our age, everybody concerned about the risks of cancer. So for those who have some dysplastic disorders that discovered during colonoscopy, probably colonoscopy at such high-risk patients recommended every 3 years. Also currently available the stool DNA testing to determine the risks for cancer. Very exciting news in the field of genomics and cancer. Lately, a couple of companies now developed what's called multi-cancer early diagnostic tests, MCED tests. For instance, University of North Carolina now routinely commercially use that test. It costs about $950. But what's important is basically it's a combination of many, many liquid biopsies that determine circulating cancer tumor DNAs, and they may predict about 50 types of cancer. And we believe that there's a great potential in that. Is just one vial of vector-tainer, and the patient can determine what are the risks for 50 types of cancer. And this completely changes the current paradigm of screenings, mass screenings, colon cancer screenings, breast cancer screenings, and that helps patients and doctors to identify the true risk for certain kinds of cancer. So genetics, diet, and exercise all play a role in dialing down the risk, but in order to become more specific, more precise, of course, it would be very nice to have data and determine what are the risk factors for cancer. Of course, very important neurodegenerative disorders, and I noticed that the whole session at this conference dedicated to neurodegenerative disorders, particular Alzheimer's disease and Parkinson's disease. So on the brain side, p-tau217 is emerging as a powerful marker for preclinical Alzheimer's disease. But it's important to use that tool selectively, not to use for the mass screening. So the doctors have to have very specific indications for using that. What's very interesting is AI powered retinal assessment also as very predicting factor, help to predict development of Alzheimer's disease at early stage. Not only Alzheimer's disease, but also certain types of dementia, especially vascular dementia, which are very prevalent at at the later stage of life. So, of course, we should not forget the basics. It's cognitive activities. Good sleep is hugely important, and controlling vascular risks of brain. So all of this put together help us to early diagnose Alzheimer's and take type measures to prevent Alzheimer's, Parkinson's disease, and other neurodegenerative disorders. So metabolic health, of course, is obesity, diabetes primarily, type 2 diabetes. So annual checks, of course, for hemoglobin A1C is very important. Of course, measuring weight and waist to determine, now we call it cardiometabolic syndrome, not just metabolic syndrome, because it's compelling evidence that metabolic syndrome leads to many cardiovascular events. So the prescription is obvious as a Mediterranean-style diet, low glycemic index foods, regular aerobic exercise, and resistance training is very important in order to reduce sarcopenia. And in the right cases, again, it's GLP agonists. Now there's new evidence, especially in the SELECT study, that demonstrated that GLP-1 agonists really reduce the risk, not only metabolic disorders, but cardiovascular disease. And just studies of a couple of months ago demonstrated that GLP agonists also reduce the risk of cancer. So we have a lot of hopes that those new generation drugs will really help to reduce the risk of many of those diseases. And it's a clear understanding and recognition that many chronic diseases are a result of dysfunctioning of the immune system. And what is very important is now the growing recognition of brain-gut axis and how microbiome affects brain through vagus nerve or through other mechanisms, through the use of incretins that synthesize in the gut. And related to that, of course, nutrition is very important, and now there's a clear understanding how nutrition affects the immune system and how it helps to improve health and reduce of many diseases. And the good news, especially fermented foods, probiotics, they really help. There's a clinical evidence that they really help to reduce the risk of many chronic diseases. But it's the research on that is still at the nascent stage of development. We really hope that together with assessment of incretins, with use of GLP-1 drugs, all of this help to improve immune health and to reduce the risk of chronic diseases. And environmental health is very important. Now there's a clear understanding that we not only have to be concerned about sedentary lifestyle and poor nutrition, we have to think about social environment of people, the psychological status, and also the clean air. And part of this lifestyle intervention should include HEPA filters, living in clean environment, and all of this together bring us to a possibility of reducing risk of many chronic diseases. So here the practical roadmap, so to speak. So we recommended that annually, now it's recommended to test for ApoB, high sensitivity C-reactive protein. It's important to perform vascular imaging, especially coronary artery and carotid artery using ultrasound assessment. Sleep is very, very important, and I'm very glad that it lately is becoming very well recognized that approximately 7 hours, but it's a myth that everybody has to sleep 7 hours. It is very personal. So somebody, for instance, 4 hours is enough for me, but for instance, 9 hours is enough for my wife, but too much sleep and too low sleep also is not good. So we have to really identify what's the best for each person. So every 3 years, for those with the risk of colorectal cancer, colonoscopy for those who have dysplastic polyps especially, recommended. And now the certain genetic and proteomics data can also help the true risk of certain types of cancer, not only colon cancer, but as I said, MCED tests really help to determine the risk and to identify early signs of cancer. So structured and flexible response to individual help with all of this. So how does this fit together? So labs, genetic imaging, lifestyle fused by artificial intelligence give us clear map of risks and timing. And there's certain limits, of course. Not every test is ready for universal use. Many of them are just like, the initial stage of commercialization. Data ethics, of course, very, very important, and they matter. And but the big picture is clear. So proactive prevention is both cost-effective and humane. So I think my time is over, but I just want to show this last slide. Is basically we are moving toward not only from reactive medicine to proactive health, but we also, the whole medicine, whole healthcare is changing the paradigm from what's called destructive medicine, which is traditional medicine we've been practicing for the past 100 years, which is based on identifying targets, destroying targets, and treating disease. It's important, but this approach is very expensive. For instance, in the United States, the cost of healthcare is about $5 trillion, which is 8 times more than Pentagon budget. The same in Kazakhstan also is very expensive, although everybody is talking about 3.5% GDP, but we know we spend a lot of time in healthcare. So the paradigm shift toward constructive medicine, it's basically, it is based on identifying personal individual resources based on the potential of immune system, and particularly in fighting cancer, especially now this PD-1 blockers, they're very successful to treat many cancers, especially melanoma, such as Keytruda, for instance. Many new drugs that help to unleash immune system and reduce the risk of many diseases and actually treat the diseases. Genetics can change by lifestyle interventions, specifically by nutrition, and here we're talking about epigenetic mechanisms. And microbiome is hugely important, and I'm glad that at NLM we have very successful microbiome lab that very successfully introduced many new methods. So altogether, it is integrated through artificial intelligence, and we're very fortunate that we live in the era of AI, and that helps to better analyze huge amount of data, biomarkers, and other personal data, and help to personalize constructive medicine, help to better analyze, reduce risk. So all together bring us to great potential to reduce risk of diseases, improve health and quality of life. Thank you so much. Okay, I realize that I'm a moderator in this session, but and I'm very pleased to present next speaker. The speakers are my very good friend Yuri Pia, but today this very important topic will be presented by Timur Lizbekov, who is associate at this great venture. So they introduced a new device called Alem that help many patients with cardiac surgery. Thank you.
Good morning, ladies and gentlemen. It's a great honor and pleasure to present University Medical Center on this stage in this distinguished conference. So we really appreciate organizers to invite us, and we would like, with a warm heart, congratulate the university with the 15 years anniversary. In contrast to previous speaker, I would talk about the final destination therapy, let's say. I have nothing to disclose. And as previous speakers said, the most stable shape is a triangle, and I'm from clinical practice part of this triangle, but I'm lucky to have my science experience and education experience, and all together are gathered with continuity. And the result may be exciting. And I would talk about one major challenge in Kazakhstan, transplantation challenges. And I would start with the world transplant mismatch. And this is the world map showing the mismatch. And I would compare the North America continent, then only 4% of population, but they perform 25% of worldwide transplants. In controversy, Africa continent is 16% of population, but they do only 0.5% of transplants. And by color, you may compare Kazakhstan and all our Asian region with Africa, where the same, unfortunately. And world transplant mismatch may be explained in these figures. So the percentage of donor organs not transplanted is 70% for hearts, it's 81% for lungs, and about 30% for kidney and liver. And ratio of unused organs to waiting list patients removed due to death or illness is 10-to-1 in heart and 32-to-1 in lung. So a huge problem in transplant world. And the current gold standard of organ preservation is cold storage, so ice. And we know that only 4 to 6 hours are safe in order to bring the organ to the transplant center. But nowadays, normothermic machine perfusion allows us to expand our donor pool. We may assess grafts. Moreover, we may do a graft reconditioning. And in North America, academic medical centers are using this technology, and almost 30% of donated organs are the procurement process is done with this technology. And results are almost same like with the gold standard cold storage. You see 3-year outcomes about 33% survival. So existing machines are shown in this slide, and the predominantly used machine is OCS, American-made device on the left-hand side for heart, for lungs, and for liver. And this system allows to perform a beating heart transplant surgery. You see, this heart was harvested from donor, was reconditioned in this system, and is beating. And surgeons will perform in non-stop or beating heart surgery. See, the heart is beating while doing heart implantation. And results are fantastic. Our journey with heart transplant program was started 10 years ago. We've done only 100 transplants, but 80% of them were possible only with this normothermic device. And we are the only one center in the region to implement this technology. And results are same like in North America, and you see in 3 years, it's about 72%. Sorry. We changed the methodology and patented several issues. And this technology is now transforming to our homemade device. See, this is a big pig model that allowed us to safely sustain beating heart in our homemade machine. And we proved, sorry for this busy slide, but the major message is we proved this concept in this 24-hour preservation protocol, and then transplanted this heart successfully. The scheme is like this, it's too busy, but that was a prototype that allowed us to sustain the organ with good parameters, vital parameters, and we proved the concept, the safety and efficacy of the system by echocardiography of the beating heart and several labs. We also may treat the heart during the preservation period by eliminating fluids and with cytosorption. So everything bad we may remove while preparing the heart to be transplanted. Also, the morphological stability of the organ was proved by coronary angiography. So it's a diagnostic tool that we may use for any organ to prove or even to treat some diseases like coronary artery disease. And by MRI, we proved stability of the myocardium, and several labs were very stable that allowed us to, I will skip several slides, that allowed us to perform a heart transplant after 24 hours of heart being in this machine. So this is the heart was weaned from cardiopulmonary bypass with low dose of supportive drugs, with good ventilator parameters and good vitals. So normal blood pressure, normal heart rate, etc. And the ALEM, meaning Astana Life Ex Situ machine, is a homemade system that allows us as a reproducible approach to sustain the isolated organs for a long term of time in near to physiologic conditions. As I mentioned prior, the OCS system from Boston allows to sustain the heart only for 8 hours. Our system allows us for multiparametric morphology and functional assessment during preservation period. Also to precise selection for transplant. It might lead to better transplant results, and it will expand the quantity and quality of organs available for transplantation. So our University Medical Center vision for this technology is shown in this slide. It is a bridge to transplant. It is a bridge to surgery, and it is a bridge to recovery for complex oncology therapy when we may excise the organ, treat it with heavy drugs and radiation, and then return it healthy to the body. And the road to organ banks is a reality nowadays, and the recent publication from International Heart, Lung, and Transplant Society is shown here, from donor center to recipient center, and the whole journey of preparation of the organ. The system was presented in Osaka, Japan at the World Expo 2025, but we think we should start, and it's our obligation to start from this region. And you know that the 75 million population region is similar in history, language, culture, etc. But also this technology may bring us to Central Asia Transplant Net for science, clinical trials, and academic medicine. So the next frontier is the bridge, to build the bridges in the region, or maybe in the world, with this exciting technology. And I would like to conclude my talk with this epilogue. Thank you very much.
Thank you very much, Timur. That was really impressive. So one comment from me is Alem, that the system they developed, means world in Kazakhstan, in Kazakh language. Okay, so that's that makes a pretty big advancing this infrastructure for transplantation. Yeah. So and now we have one more speaker. Not the last, but it's not the least. So it's our keynote speaker for today's morning session is Professor Bart Loijs. He is from Antwerp University Hospital, and he's our major collaborator in heart projects. Yeah, please.
Good morning, everybody. Thank you for the introduction, and it's my pleasure to share this celebration and to be part of exciting life science. I especially want to acknowledge Nur for inviting us and the nice collaboration that we have with Belgium and Antwerp. So it's my pleasure to talk about my favorite disease, which is inherited aortopathies. Aortic dissection is a major healthcare problem, and it's the cause of death of about 1 to 2% of the population. And it comes basically in two flavors. It can either affect the thoracic part of the aorta, which we call TA, or thoracic aortic aneurysm, or the abdominal part, which we call AAA, or abdominal aortic aneurysm. And although the two exist, they are quite different in their genetic architecture, in that sense that abdominal aortic aneurysm is mostly linked to risk factors, classical cardiovascular risk factors. And although some genetic influence has been identified, there is not a single monogenic cause of abdominal aortic aneurysm. And this is in sharp contrast to what we know about thoracic aortic aneurysm, where we see that about 1 out of 5 patients does have a positive family history, and quite often there is no other risk factors. And over the years, many genes have been identified, either in syndromic forms where other organ systems are involved, or non-syndromic forms. So if you look at this pie chart, you recognize the 1 out of 5 familial cases, and of those, about 15% are non-syndromic, which we call familial thoracic aortic aneurysm, meaning that there is no other organ involved than the aorta. And then there is syndromic forms, which I will go a little bit more into depth today. But there's also a large group of sporadic cases, well, 80% that do not have a family history. The fact that this is an important disease that can be tackled by prevention comes from the observation in populations where they looked at the relative risk for family members in patients that present with aortic dissection. And there's 2 quite recent big population studies, one from Taiwan and another one from Denmark, and both describe high relative risk or hazard ratios for the family members. So it indicates that there's strong evidence to do familial screening to go into the genetics of aortic aneurysm and dissection. So this was also picked up in the recent guidelines from the ESC. For the first time, it's really acknowledged that the cardiologist and the cardiac surgeon should take a family history, because it's a very important way to identify risk for family members, and also for the first time in the guidelines, they point at the importance of cascade testing. So if you identify a mutation in a proband, it's very useful to go for the family members, because most of these conditions are autosomal dominant, implying a 50% recurrence risk for having the pathogenic mutation in the first-degree relatives. But still, how do we define which patients we will send to the genetic centers or the expert centers to evaluate genetic risk for aortic aneurysm? They have drafted this quite complicated scheme, which I think we can basically summarize in 3 rules. And so that genetic testing and additional imaging of first-degree relatives in an expert center is indicated in all patients that have a dissection above 60 years, if the dissection is without hypertension, or if there is also hypertension, there should be an additional risk factor. And I'll list the additional risk factors on the next slide. There is an indication to do this in all patients with an aortic aneurysm that has a Z-score, which is a standard deviation without hypertension. And again, if there is hypertension, there should be an additional risk factor. So what are these additional risk factors that we should look for? First of all, it's the family history of certain cardiac death. So that's where the importance of the family history comes into, of certain cardiac death below age 60. The presence of thoracic aortic aneurysm, of course, and dissection in the family, but also the presence of peripheral or intracranial aneurysms is considered as an additional risk factor. And then there's also what we call connective tissue findings that may point more towards syndromic forms. For Marfan syndrome, we think about pectus deformities, the presence of joint laxity, pneumothorax. Specifically for Marfan syndrome, you look for the disproportionate tall stature with the long fingers, as shown here with the positive thumb and wrist sign. And you do a slit lamp exam to look for lens dislocation, which is present in about 60% of the Marfan patients. Or you can look for soft markers for the Lewis-Dietz syndrome with widely spaced eyes, the bifid uvula, which is basically considered as a minimal expression of a cleft palate, or on imaging presence of tortuosity. So these factors can help you to identify those patients that would benefit from genetic testing. It's not always easy to interpret these. I just want to point at some tools that we're developing within the European Reference Network of ASORN. They're freely available online, where for each gene that's identified in the world of aortic aneurysm and dissection, we provide specific guidance on how to interpret these variants. So there's, with the advent of next-generation sequencing, a high number of genes that have been identified. I think the current count is around 44. Here is the long list. I don't have time to discuss all of them. I just want to point at some observations. Despite this long list, only less than 30% of the probands we can at present genetically explain. We've also seen that the more recently identified genes only explain small proportions, usually less than 1% of the whole TAT population. And another problem from a genetics perspective is that the more recently discovered genes have a higher degree of non-penetrance, meaning that you can identify family members that will not develop the disease, and it also makes it more difficult to identify these genes in those families. So if you look at the overall picture that comes out of the knowledge that we gather from identifying these genes, we can say that there is 3 major classes of genes involved in the pathogenesis of aortic aneurysm dissection. The first group is related to the presence of mutations in genes that encode for proteins that are important in the extracellular matrix. For example, fibrillin-1 in Marfan syndrome, they are believed to give structural integrity to the aortic wall, but I will also point at more recent evolutions that we know that they do have other roles beyond the structural integrity. A second major group is what I will also talk about, genes that are involved in the TGF-beta signaling. So through the interaction of integrins, the cell senses what's happening in the extracellular matrix and drives this signaling pathway, which in its place then will again tell the vascular smooth muscle cells in the aortic wall how to behave, and leads to changes in the function of endothelium, and in the end also extracellular matrix remodeling. And then the last class is genes that encode for proteins that are important in the contractile apparatus of the vascular smooth muscle cell. The most well-known one is actin. So I'll spend a little bit of time on this last group. Most genes that are in this a group of vascular smooth muscle cell contractile apparatus partners will lead to what we call non-syndromic forms. So usually you will only find aneurysm as the presenting sign or dissection. In myosin heavy chain related disease, you can see an association with patent ductus arteriosus. In the ACTA2, sometimes you also see premature coronary artery disease and premature stroke, but it's not obligatory. And PLK-J1 is often associated also with hypertension, showing that we are also having a multifactorial pathogenesis where other factors can come into play on top of a baseline genetic predisposition. So most of what we know about aneurysm development and genetics is derived from a study of rare monogenic forms, with the most well-known one, Marfan syndrome. So, as I told you, it's a deficiency of fibrillin-1. You can find the skeletal eye and most importantly the cardiovascular findings where the aneurysm typically occurs at the root. You can also have mitral valve disease. It's autosomal dominant. 30% of the patients are the consequence of a de novo mutation, so there should not always be a family history. And what we learned is that this is really a monogenic condition. With current technology, we're able to identify the causal mutation in about 95% of the patients. There's a wide range of genotypes, and there is very little really strong genotype-phenotype correlation. So meaning that from the nature of the mutation, you can not always predict outcome with regards to aortic involvement or treatment outcome. There's one exception to that, is that if a cysteine amino acid is involved in the mutation, there's a threefold higher chance that they will have eye problems. But again, it's not absolute, it's just a correlation. So the most important progress that has been made in understanding the development of aneurysm is the fact that it moved from a disease where we believed it was a structural problem to a condition where we do know that the extracellular matrix also has a role in regulating a local hormone, cytokine, TGF-beta. So in a normal situation, TGF-beta is sequestered to the extracellular matrix through the interaction with the latent complex. And in a pathogenic situation, this cytokine is dysregulated, overactive, and drives a lot of the phenotypical characteristics. This was shown through a lot of work, mostly based on mouse work in Haldiz's lab, where they could show that if you block the overactivation of this TGF-beta pathway, you can rescue the phenotype, at least to a very strong extent in the mice. So this led to the idea that maybe we can block this overactivation and try to rescue the phenotype also in humans. Because if you have to restore a structural protein, it's much more difficult to accomplish. So from knowledge in the nephrological world, kidney failure, we do know that angiotensin receptor blockers, such as losartan, have an effect on this cytokine. It brings down the total amount of TGF-beta, but also the amount of active TGF-beta. And it has cross reaction with the angiotensin pathway, preventing this overactivation of the TGF-beta signaling pathway. So a big trial was set up, and we compared the, at that time, standard care of treatment, which is the beta blocker, with losartan in a big population of 600 individuals between 6 months and 25 years old. This group was chosen because the growth there is still most pronounced. But unfortunately, the study did not show a difference between the two treatment regimens, saying that both are effective, but losartan not being superior as we had seen in the mouse models. There might be some explanations for that. I'll come back to that. But for example, the dose of beta blocker that was used in the study was very high because the request of an up titration to the heart frequency, which was not done for losartan. So on average, I think in a pediatric clinic, patients would get 1 milligram per kilogram nowadays. In this study, a lot of patients went up to 4 milligram. So one lesson that we did learn from this trial is that early initiation of therapy has a beneficial effect. So the delta Z of the aortic root growth was more effective in the younger age groups. You can see it both for atenolol and losartan. So this is an important lesson, but the question is what would have happened if we would have used a very high dose of losartan or an angiotensin receptor blocker, which we didn't do in the study. What about the combination? It was also not studied, and since then also a lot of studies have been carried out in other populations. Can we look at these questions with doing a meta-analysis? So the dose does matter. This is an experiment in a Marfan mouse model where you either gave a placebo in red, medium dose propranolol, high dose propranolol, low dose losartan, medium dose losartan, or high dose losartan. You can clearly see that also increase of the dosing of the angiotensin receptor blocker brings down the aortic growth to the wild type level or even better. So this seems to indicate that high dose would be more beneficial also for the sartans. This was studied in one study in the UK where they showed that with a high dose there was indeed a 30% decline in irbesartan. In this study, they saw no signal for interaction between irbesartan and the beta blocker. So Alex Pitcher from Oxford did a big meta-analysis looking at all the trials, and many have been carried out in different countries. And they indeed confirmed that both the losartan and the beta blocker are effective in slowing down the aortic growth, and that there might be some indication, but none of the trials was really designed to prove this for combining the treatment. So we still have work to do. We can slow down the aortic growth, but we cannot stop it at present. So one of the very important questions that we try to tackle is how can we explain that even within a family where they have the identical primary mutation, being it fibrillin-1 in the Marfan patients, there is variability in the severity of the phenotype. And one of the ideas is that we can learn by doing genetic studies, looking for genetic modifiers that determine the outcome with regards to the aortopathy severity. So one way to do that is to perform traditional old-fashioned linkage analysis in which we don't look for the primary mutation, but we try to identify the modifiers. So you identify large families with variability in cardiovascular severity, and you perform a linkage analysis with the severity as the phenotype, and then try to see if we can identify the genes that explain this variability. So this is what we did. We identified these families, stratified them as patients with severe aortic disease or no aortic disease or very mild aortic disease, then do the linkage. And we identified a linkage peak that showed that all the disease status, so the mild aortic disease was linked to the presence of a specific protective haplotype. And then you can look under this linkage peak on your genome, which genes are there, and as such, we identified a potential protective modifier, MAP3K4. Maybe the name doesn't tell you anything, but it's linked to the TGF-beta signaling pathway. You have a canonical and non-canonical part, and this gene is exactly downstream in this ERK non-canonical pathway, TGF-beta signaling pathway. You can do very similar things in mice. It's very striking that depending on the background of the mice, an aneurysm can be present or not. So this is an example for Marfan syndrome. If you have the mouse on a black 6 background, you develop aneurysm, but on the right you see the echo with the yellow arrows pointing at the sinus with huge aneurysms on the 129 background. So this is here reflected in the graphs, and it even affects the survival in that these 129 fibrillin 1 deficient mice go on to develop dissections and will die, whereas that does not happen in the black 6. So again, you can do the same as we did in the humans and look for the extremes, identify the mice with huge aneurysms, the ones without, and do linkage analysis, identify the regions on the genome, and identify the modifiers. Just want to show here that you can clearly link the presence of these modifiers in an epistatic way, so having 2 of them is beneficial in the black 6, having none of them in the 129 background gives you big aneurysms. And again, those genes map to the same pathway, saying that this pathway is an interesting pathway to be tackled in the treatment. Is this exceptional? No. For almost every mouse model that we have now in aneurysm, we can see protective and aggravating backgrounds in the mice, so giving us lots of opportunity to try to map modifiers. So this TGF-beta signaling pathway jumps out. It was discovered after we did some clinical observations. At the time I was a fellow at Johns Hopkins, where we saw Marfan-like patients that did not have a fibrillin 1 mutation, that have the widely spaced eyes, the bifiduvula, craniosynostosis that distinguished them, and we identified that this is a different condition with a different natural history, where there is earlier surgery needed because they dissected smaller diameters, and there's also more widespread prevalence of aneurysm because the aneurysm is not limited to the sinuses as in Marfan syndrome, but spread throughout the arterial tree. So what did we learn about a molecular basis? So we identified first mutations in the receptors for this cytokine, the TGF-beta receptors. They are predicted to lead to a loss of function, but there is some paradox in that if you have a complete deletion of the gene, it does not cause an aneurysm phenotype. If you do that for TGF-beta receptor 1, it even leads to a cancer phenotype and not to an aneurysm phenotype. So it suggests that you need some residual protein left in order to cause the aneurysm. So despite the predicted loss of function, when you do a staining for a marker for activation of the TGF-beta signaling, which is phosphorylated SMAD2, you can see that in the aneurysm dissection phenotypes, there is increased presence of this marker. So it leads to this paradox where in Marfan syndrome we have overactivation due to the fibrillin 1 deficiency, and in Lewis-Dietz you also have overactivation due to the loss of function of the receptors, but they both lead to increase in overall signaling in the aortic wall. So this was confirmed in mouse models, and we can reproduce the aneurysm, the tortuosity, and there is a hypothesis that still needs to be proven, but we think this paradox comes from the fact that in the aortic wall you have lineage-specific contribution of cell types, so meaning that the embryonic origin of your vascular muscle cell determines how it will react to the TGF-beta overactivation. So by now we have identified many genes in this pathway, both at the receptor level, cytokine level, downstream effectors, and we are moving towards a world where aortic aneurysm management becomes gene-guided. I will give some examples of that. For example, when you look at the genes that are involving the contractile apparatus, PRKG1 clearly has a worse prognosis than the other genes, and as such justifies earlier surgical intervention. We can do the same exercise for the genes in the Lewis-Dietz pathway, where we can shift the threshold for surgery according to the gene. So that's at the gene level. Sometimes it is very specific nature of the variant that determines the outcome. For example, in collagen 3, where you have vascular EDS, if you have a null mutation, so meaning less quantity, it's much better than having worse quality, and it also determines the outcome with better outcome for the patients that do have less quantity. Sometimes you can really point at very specific gene mutations at specific spots. So this is an example in ACTA2, where this arginine at position 179 has a childhood onset, very severe, very similar in TGF-beta receptor 2. This arginine at position 258 has a much worse outcome. So really moving into having the gene information as our guide to do the management. Of course, I told you that we still miss a lot of genetic explanation. This could be to more genes, of course. I think genome and exome sequencing will help. But we should probably also start to look more into single nucleotide polymorphisms, so more common variation that can also contribute to aneurysm, and there is an example of that in fibrillin 1. Copy number variation is, there is a nice example of a duplication around myosin heavy chain that gives you a 12-fold risk increase for dissection. And there is the whole world of non-coding variation that probably will also help to solve this problem. So this is how the genetic architecture looks like at present, I think. We have pure monogenic forms where just the presence of the gene is sufficient to cause the disease. There's probably a subgroup that is more polygenic, where there's many variations in the genome that combined with maybe lifestyle factors can cause the disease. And then there is a group in the middle where you have what we call the class 3 variants, the variants that we don't really know what they mean, as predisposing together with or without additional environmental factors. This is becoming into clinical practice. There was a very nice paper last year in European Heart Journal where they tried to combine genetic risk scores with classic clinical factors in medical history. And for example, they were able to have an area under the curve when they combined these risk factors to predict an aortic size of more than 4 in nearly 84%. So that's getting to a place where we can try to think about using it in the clinic, combining classic risk factors and genetics. And I think that's where the field is moving. There's other evolving evidence that we should not only look at the diameter, but try to bring in other factors such as altered flow could be predictive along with the genetics. The flow predicts to a certain extent also the growth rate. Other people have looked at the tortuosity as a marker, additional predictor for severity. This is also reflected in studies done in Marfan syndrome and Lewis-Dietz syndrome, where if you have this tortuosity, there is a odds ratio of almost 4 for the risk for dissection. This is similar in Marfan syndrome. Another factor that's related to that is lengthening of the aorta. So you can start to combine all these factors and build a risk model that's not only relying on genes, but also on these dynamic factors and the emerging risk factors such as the lengthening, the tortuosity, stiffness, and biomarkers. I will stop here by saying that we have lots of data at our hands now. I think, as mentioned earlier today, AI will help to uncover the correlation and the epistatic effects between those. We really need good deep phenotyping to build this integrated model where we bring in additional imaging biomarker findings. And this can be done with studying also iPSC models and mouse models. I would like to thank the people involved in the lab, a very enthusiastic group of young researchers, and the people in the clinic, and the cardiology and cardiac surgery department from Antwerp. And I would like to thank you for your attention.
So we still have time for a couple of questions because we pushed. Is there any questions? Any questions, please? We have a few minutes before coffee break. So actually, Bart was very shy. Bart was but the syndrome that he was describing actually was giving name recognition of the major discovery contributor to this discovery of this syndrome. Actually, it's Lewis-Dietz syndrome. Correct? We greatly appreciate it. This is your work. Thank you. And a couple of more. So it's time for coffee break. But if some our participants who would like to have a translation, please, the headphones are available. So translators are working. So please don't be shy. So thank you. So we have to get back here, at 11 a.m. We're back to our schedule as we planned. Thank you. See you.
Check.